Hello, everyone, and thank you for taking time to attend my presentation today. I'm so thankful for the invitation by the SMALP-Journal Club to speak with you all. Thanks again for having me. To introduce myself, my name is Chris Sander. I'm a graduate student at Case Western Reserve University in the labs of Professors Philip Kaiser and Chris Balcheski. We recently published a paper entitled Nanoscale Resolution of Native Retinal Rod Disc Membranes Reveals Differences in Lipid Composition in the Journal of Cell Biology. And I'm happy to be able to share that story with you all today, as well as answer any questions you might have about the work. Let me first begin by describing the impetus for this work, though. We study proteins in our labs involved in the process of vision, which includes many membrane proteins, making it very important to learn about the SMALP technology. So there we go. So biochemical conversion of light to electrical signals begins in the back of the eye in the retina, more specifically cells called photoreceptors, and in their outer segments. Photoreceptors depend on distinct membrane compartments to support their specialized function, namely rod outer segment discs, or ROS discs, as you can see here in this EM image. Further segregation of these compartments has been demonstrated between the center, where rhodopsin begins phototransduction, and the rim, where ABCA4 facilitates used chromophore clearance, and another protein called peripherin ROM complex maintains the membrane curvature. And under normal conditions, light causes the covalently bound 11-cis retinal to photoisomerize to all trans retinal, as seen here, and leave the rhodopsin protein and is then released into the membrane. And this happens after rhodopsin has given the signal that light has hit the retina to the G protein transducin. Once all trans retinal is in the membrane, it can form a reversible Schiff-base linkage to a molecule of phosphatidylethynolamine, or PE, which is recognized and flipped out to the cytosolic leaflet by ABCA4. In pathogenic cases, however, of mutations to ABCA4, all trans retinal PE accumulates in the inner leaflet along with increasing amounts of free all trans retinal, and that leads to the formation of toxic amounts of all trans retinal and bisretinoids. Mutations in ABCA4 are known to cause the leading form of juvenile blindness, Stargardt disease, that robs children of their vision, many becoming legally blind by adulthood. Unlike protein localization, identification of critical differences in membrane content has not yet been expanded to lipids due to the difficulty of isolating domain-specific samples. Determining the lipid content of certain domains of a continuous ROS disk could yield therapeutic insight, as protein-lipid miscommunication is a known pathological mechanism that may extend to diseases like Stargardt disease. To this end, competing hypotheses have been put forward as to the likely role of very long-chain polyunsaturated fatty acids, or VLC-PUFAs, as shown here. The level 4 is responsible for elongating long-chain PUFAs into VLC-PUFAs, and mutations of it cause what is known as Stargardt 3, sharing a similar phenotypic profile as ABCA4-derived Stargardt 1. If VLC-PUFA biogenesis being disrupted causes a similar phenotype to ABCA4 disruption, perhaps a healthy interaction between ABCA4 and VLC-PUFAs is disrupted in that Stargardt 3 disease, causing destabilization of ABCA4. So early reports suggested that VLC-PUFAs bound tightly to rhodopsin in the center of ROS discs, perhaps surrounding the transmembrane with their long polyunsaturated fatty acid tails. Another hypothesis suggested that the similar phenotype seen in ABCA4 and the level 4 mutants may tie the etiologies together, as stated in this particular review paper. So the pertinent questions then that we set out to answer were, where do VLC-PUFAs reside in the ROS disk, and is the rim membrane environment unique compared to the center of the disk? And so the biggest problem in addressing these questions, as I alluded to earlier, is somehow fractionating the disc membranes while remaining the native lipid profile. One fact that we tried to leverage is that the Ross discs have this amazing separation, as you can see here, of rim proteins from the center to the point where there's no detectable ABCA4 or peripherin complex in the center of the disc. And we know this from EM images of immunolabeled single Ross discs. What we planned to do was use SMA to co-purify membrane proteins and their native lipids from the rim and center regions of photoreceptor Ross discs, and then subject those to those co-purifying lipids to untargeted lipidomic and fatty acid analysis. We were confident that doing all these steps of sample prep on ice or at 4 degrees would be sufficient to keep the exchange between SMALPs at bay. There are several reports in purified SMALP discs of homogeneous and also mixed lipid cases where there was a rapid swap of lipids between nanodiscs. So to further convince ourselves that it wasn't just that doing this whole procedure at 4 degrees or on ice was enough to keep that from happening in proteolipodisc circumstances, we hypothesized that we might see, we hypothesized that if we purified 2 samples from the same region, both ABCA4 and peripherin ROM, that are known to be distinct from the center, that we might have evidence that the differences between these hypothetical differences between these 2 samples and the center would be more in line with one another than, say, between rhodopsin and the rim samples or rhodopsin and the peripherin samples. And so we did this basically as a way of convincing ourselves that post-hoc sorting after solubilization wasn't going to be happening. And we further thought that this was a good test case because peripherin ROM1 and ABCA4, both of the components of this complex, have very low sequence identity to ABCA4, with I believe 16% between peripherin and ABCA4 and 22% between ROM1 and ABCA4. And so there was little reason to think that ABCA4 and peripherin ROM could both preferentially sort the same lipids and generate the same differences from rhodopsin in nearly identical ways. That sort of explains the experimental setup that we used. In order to purify these things, we used an established antibody against rhodopsin called 1D4. We also designed an antibody against ABCA4 for better preparative scale purification, which was then followed up because we still had some remaining contaminants. We used size exclusion chromatography to further purify that protein. These are all Kumasi gels, by the way. And then we also collaborated with the Steyert group in Belgium to screen their nanobody library that has been published for one that could bind and purify peripherin ROM together. And you can see here that we got a nice pure population of peripherin and ROM together, and the corresponding nanobody eluting with the protein. So the resulting SMALP proteins, as has been reported before, retain ligand binding abilities, and as you can see here, ABCA4, an ATP binding cassette protein, was able to bind in a saturable manner to ATP, as you can see here in this tryptophan quenching assay. And regarding rhodopsin, we were also very encouraged by seeing that the purified in the dark rhodopsin, which has a characteristic absorbance peak when it's bound to that molecule of 11-cis retinal, it gives a characteristic absorbance peak at 500 nanometers. And when bleached or exposed to light, for in our case, bright white light at 7 minutes, we were able to completely remove that chromophore. So we bleached the protein, and then if we add back more retinal, cis retinal, we're able to reform that complex. So we create another dark state protein, and that was stable at room temperature for many days, which was very surprising to me because this process generally isn't possible in detergent, let alone the stability that we were able to see in SMALPs. So that was great to see. The result of our lipidomic analyses, which was done by the Acylcarnitine group, as I said before we started recording at the Salk Institute, gave us many points of distinction between the rim samples and rhodopsin, a trend that was confirmed using principal component analysis, as you can see here. We were struck by the panoply of components extracted by SMALPs and measured by the lipidomic analysis. Many membrane components co-purified with the sample proteins, including lyso-phospholipids, sterols, sphingolipids, acylcarnitines, free fatty acids, cardiolipin, mono, di, and triglycerides, and some more species as well that were all covered in the supplement of the paper. Our comprehensive report of the components of Ross disc membranes across figures 4 and 5 of the paper and also in the supplement was, to our knowledge, the most varied of any tissue extracted by native nanodiscs so far, although I'm sure that this community is rapidly making that statement obsolete. One of the many differences was that the rim seemed to be relatively lower in PE and had a commensurate increase in PC when compared to the center of the disc. So we noted that as well. Differences in content of acylcarnitine and lyso-phospholipids were the most notable in the analysis of the lipid classes. And while carnitine has been reported to be in ocular tissues before, our data further localized at least some of the acylcarnitine to the center of the Ross discs. Previous work has shown that injection of carnitine into the eye can be protective in an ocular hypertension model. Our results, which place acylcarnitine in the immediate vicinity of rhodopsin in the membrane, suggest that carnitine may act as a check on normal oxidative stress in the outer segment disc membranes. To that extent, or to that end, supplemental carnitine could increase the protective effect afforded the retina by endogenous levels of carnitine in the outer segment discs, but more evidence is likely needed to confirm this. The presence of lyso-phospholipids has also been reported at the tissue level in bovine and human retinas as a whole, but their specific functions in the retina are yet to be determined. As a surfactant, though, lysophosphatidylcholine, or LPC, has been shown to increase membrane fluidity, which is itself important for protein reorganization in the discs, especially with the frenetic activity that we see with rhodopsin signaling. Our data show unequivocal differentiation of the lysophospholipids, with short saturated species in the rim samples and longer, more polyunsaturated fatty acids in the rhodopsin or center samples. It's in addition to that general effect of possibly acting as a pseudo-surfactant or something that gives an increase in flexibility to these membranes, or lateral diffusion in these membranes. It's also conceivable that the lysophospholipids in the center of Ross discs interact specifically with the membrane proteins in a signaling capacity. Lysophospholipids have been shown to interact with GPCRs to initiate G12, 13, GQ11 family G proteins, as well as inhibitory and stimulatory G proteins, thereby affecting various downstream intracellular signaling pathways. So the PCA loadings from the aforementioned analysis suggested to us that the saturation level of the lipid species might be playing a role in separating these two regions. As you can see here with a rank order list of PC1 scores from +1, which was near the rim samples, to -1, which was where rhodopsin clustered. You can see that there are many more polyunsaturated species in the rhodopsin clustering area of principal component 1, and many more saturated fatty acids in these rim sample regions. And so when we hydrolyzed the head groups of all of the lipids and only considered the fatty acid chain length and saturation, we saw even more striking differences between the rim samples and rhodopsin. And what became apparent was that the rim is highly enriched in short saturated fatty acids like palmitate and stearate, and rhodopsin samples were enriched in long and VLC pupas compared to the rim samples. These results, we feel, give strong evidence that native lipid compositions are retained in the extraction and purification procedure if it's kept cold, as we did here, and as is done in most protein purification procedures. In our results, we don't have just 2 or 3 instances of matching differences between the rims samples and the center, but we have over 20 examples of statistically significant differences between these 2 samples and rhodopsin. So we believe that's pretty strong evidence that we can use SMALPs to differentiate between continuous membrane regions. Therefore, we were able to answer a basic scientific question that had eluded study for decades with a small bit of biochemical ingenuity, thanks to SMA, of course. We now know that the rim region is largely devoid of VLC pupas, which appear to localize with DHA and the other important long-chain fatty acids near rhodopsin. This means that the etiologies between Stargardt 1 and 3 are likely separate, as a loss of VLC pupas is unlikely to impact ABCA4, at least where it resides in the mature Ross discs. In this process, we also learned that the center is more unsaturated, contains nearly all of the LC and VLC pupas, and is relatively enriched in PE, among many other differences that I wouldn't want to spend time on in this format, but are formally discussed in the manuscript. And if you have any questions, I'll be able to cover those. I have all of the figures in post-acknowledgement slides. But anyway, I'd just like to thank you all for your attention today. Special thanks as well to the members of my lab that were instrumental, or my labs, the labs to which I'm a part. I'd also like to thank both of my mentors for their frequent guidance and encouragement. Obviously, I couldn't. No PhD student does this alone. I'd also like to thank, in a special way, Professor Skowronski-Krachik, who had essential contributions to this story and has also given me constant advice, which has been much appreciated. I'd also like to thank the Psychatlian Lab, the Salk Institute for handling the lipidomic data collection, and the Steyert Group for their help in developing a nanobody against peripherin. Thank you also to Polyscope, in case anybody here, I don't know if anybody here works for them or something, but they sent us samples of the 1-to-1, 2-to-1, and 3-to-1 SMA back in 2018 when we started doing this and playing around with it. So that was instrumental in this project that we thought was very exciting, and it's also a very large part of my dissertation. So I thank them very much for those kind gifts, and I'd be happy to take any questions that anybody here has.
Yeah. How did you separate the rim area from like the extended area? I mean, rhodopsin-enriched area.
Right. Yeah. So the basically our plan was to solubilize all together. So what I do is pre-purify these by snapping these cells at this extended primary cilium region called the outer segment of rods. We're able to pre-purify this part, which contains all these discs, and then I solubilized all of that material together. So I generate essentially the rhodopsin SMALPs, peripherin ROM SMALPs, and ABCA4 SMALPs, and then preferentially, or then enrich to purity each one of those components, and then we did just essentially a Bly and Dyer extraction, chloroform methanol extraction of the lipids from the SMALPs, and then subjected those to untargeted lipidomic analysis. So there wasn't a pre separation between the center and rim. It was all done through purification of these proteins, immunopurification of these proteins.
Excellent.
I'm not sure it's necessary to use immunoaffinity. We did it because we had the resources to do that for this project, but the same sort of thing could be done biochemically if there was something about your target protein that was biochemically isolatable from native sources. That's the great thing about these SMALPs, is that you're able to do these studies in native systems.
Excellent, thank you.
Mhm.
Any other questions?
I have a question.
Sure, please.
Hi, Chris. Long time no see. It's Take.
Oh, hey, Take. How are you?
Yeah, good. And my question is, it's beautiful work to see the environmental lipid composition, but is it possible to do the same thing with detergent to see the functional lipid binding directly associating to the protein, or you never tried it?
Yeah, so I mean, if you're looking for very tightly binding
Yeah, exactly.
phospholipids, I think that kind of thing could be done. And I know that there have been even some crystal structures with co-purifying and very tightly bound lipids. This was more interesting to us for the reasons of looking at very tightly binding and non-annular and annular lipids that were roughly within the radius of the nanodisk. So each one of these using the 2-to-1 SMALP, and also based on our little negative stain study we did with this, something around 11 or 12 nanometers in diameter. So we're going to get more lipids than if we were using just the ones that preferentially come with detergent. We also get rid of somewhat of the complications that maybe only certain types of certain charge maybe, or something like that, or certain chain length would be amenable to solubilization with detergent coming along with the protein. So we get rid of that problem a little bit using this method as well. But yeah, if there was a very tightly bound or necessary fatty acid or phospholipid, I could see that coming along with it.
Yeah, okay, thanks.
ABCA4 is sort of in the visual cycle. It's not directly involved with signaling with rhodopsin. So rhodopsin will, as a GPCR, interact with the G protein called transducin and transmit the signal of phototransduction that way. And that signals through another protein called PD6 or phosphodiesterase 6 that then interacts or changes the concentration of cyclic GMP in the cell, leading to the closing of cyclic nucleotide-gated ion channels on the plasma membrane, and that leads to depolarization of the cell, or rather hyperpolarization of the cell, and then the release of that is how we get the electrical signal that passes on to the rest of the neurons in the retina to the brain. So the ABCA4, instead of being involved directly in that process, it's just right now the number one theory as to what it's doing is helping reduce the amount of all-trans retinal buildup in this inner leaflet that's inaccessible to this retinal dehydrogenase that is responsible for turning retinal into retinol. And retinol is much less reactive and poses much less of a threat to retinal health than the retinals, which can form these bisretinoids and also just interacting on their own and reversibly interacting with the opsin. It could lead to aberrant signaling, that kind of thing. So making sure that it's accessible to these retinal dehydrogenases is thought to be what ABCA4 is doing. It's just increasing the and as far as the like a protein-to-protein interaction with ABCA4, that hasn't there is no known protein-protein interaction between those two. So I think that's why we're able to separate so nicely in SMA and in these purifications. I know that in the let's see if I have that. Yeah, so in this purification where I still have remaining protein that seems to be at the level of rhodopsin here, I believe this is a contaminant just because there's so much starting material that's rhodopsin that it sort of binds to the column itself or to the beads itself, and that's why we use size exclusion to separate the SMALPs that had rhodopsin from the SMALPs that had ABCA4. It's not that rhodopsin was in the SMALPs with this and co-purifying as a complex.
Excellent, that's so beautiful. Is there any reason that ABCA4 is specifically localized in the rim area? Is it because of the lipid, or there are some regulators?
Yes, that's a great avenue for future study. One of the possible reasons is let me see if I can pull up so in this negative state EM structure, you can see that there's this very large and for the it actually extends beyond where this EM structure was, actually not in this. So this is a very closely related protein, ABCA1, which is also a lipid flipase. We use that to model the size that we should see in our EM structures. And but basically this extended intradiscal or extracellular domain needs a lot of room, and the discs are stacked in such a way that most likely the only way that this can fit is to put it into the rim of those discs. So that's partially a reason for why it's there, but it could also be a function of preferentially seeking out those that lipid environment that peripherin and RUM also seek out. But I think it's much more likely that it's this protein steric requirement.
Excellent, thank you. So beautiful. Who is working on the structure of ABCA4 now? Is that your group or?
Oh no, there was a group. They were in New York that just published in this past year structure of ABCA4 via cryo-EM. It's incomplete in this region up here where the ABCA1 has a little bit more information still. That's why I use this as a size comparator here. So many of the residues in the center of the protein were very well resolved. So And so they did that in a nucleotide bound, so a non-hydrolyzable, or yeah, I believe non-hydrolyzable ATP, and also APO form. So yeah, it was nice to see, because we've been working on it and had struggled with it's a pretty hard protein to work on. It's very large and very much doesn't like to be kept soluble. So SMA helps greatly in biochemical assays, but as far as cryo-EM, it's a different story in making these things concentrate well and also display themselves nicely in the thin ice. So
excellent, thank you very much. Any other questions? Very exciting questions you address in this paper. That's something that's distinguishes your group from others. You know, you had a biological question and you use SMA to answer those. That's beautiful. That's really good.
yeah, thank you. Yeah, we definitely wanted to. We didn't want to just provide a test case, but also answer something biologically. So that was certainly involved in the impetus for the study. But yeah, we also and we had prior to submitting, we had thought of it, very early in the project. We had been thinking about this idea of this rapid swapping of lipids. Would this even be a useful method to use to distinguish the lipid differences between the two regions? And luckily, we had this perfect case where ABCA4 is here, peripherin ROM is also in the same region, and there's really no good reason, sequence-wise, that they should have similar lipid profiles, and especially that they should have identical differences between the two of them and rhodopsin. So it was just a really nice test case, and I believe that it's explainable through the fact that this entire procedure is done as most protein purification is done at 4 degrees or on ice. So most of these lipids aren't at the temperatures required for that rapid exchange, as was, and in those papers that did show rapid exchange, I know that the temperatures used, the lowest was 10 C, but most of it was done at 25 or 30. So while certainly biologically speaking, if there was some biological implication for which I'm sure there is for these SMALPs, then that would be an important thing to know. But if you're trying to use these for structural studies and you want to claim that there's a native lipid environment, I think you're well within your right to do so, given our results.
Excellent. And I'm not sure how many immunopurification with the SMALP has been done in the literature, but maybe other people can comment. But this is a very good immunopurification of using the SMALP. You know, that's normally a lot of people use tags. So
right.
You are able to purify endogenous complexes using
right.
That's amazing.
Yeah, the problem most of the time with that is that if it's exogenous protein, then you're likely not expressing it in the native membranes. So I know a lot of people try and find a similar expression system, like that would have a similar lipid profile or something like that. But really, if you have a system amenable to where you can get the large amounts of the organ that you're studying, and you can use these SMALPs. It was extremely well solubilizing of the retina tissue. I don't know if that carries over to if you had, I don't know, mouse brain, for example, if you're working on some proteins expressed in the brain or something like that. I know that they're, especially in neuronal studies, this might be interesting because of the highly specified lipid environment that usually calls for. It was just really exciting. I'm very thankful to the people in the SMALP community for coming up with this technology and recognizing its worth too.
Excellent. So, did you have any issue eluting your protein from during your purification with antibodies?
I'm sorry, I missed the last part of that. If you could just speak it like a little louder.
Sure. Did you have any issue eluting your protein using, during your immunopurification?
Oh, so as far as elution, no. We were lucky enough that in the in the case of peripherin ROM complex, this nanobody actually has a his tag on it. So we were able to use a his tag based purification system, a nickel NTA system for purification of that complex in the SMALP with an antibody. So in that case, that was relatively easy to elute, just using the normal imidazole. And then, there. And then in the other cases of rhodopsin, we used the epitope peptide to elute, and then that likewise for immunoaffinity with ABCA4 elution, we just use the known epitope peptide to elute those. So.
Perfect. This is beautiful. Yeah.
Yeah, no, that might be a problem if you're purchasing if you're doing this at small scale and you have an antibody that you don't know the epitope or you don't know the paratope rather. Then you're going to you'll have a problem.
Excellent. Normally, when we use a small or any type of polymer-based purification, we are concerned about the yield as well. So you didn't have any issue with the yield, or did you optimize this procedure?
Oh no, we had actually increased yield as compared to in detergent, or at least the detergents we were using. I think I can, yeah. So this is just the in this head-to-head case with element G, we had about half of the Ross membrane proteins were left in probably a little less than half, but in ABCA4 it was roughly half. The amount was left in the pellet, so that's just all protein that we don't get to use now. So the amount left in the pellet was significantly less with using SMA. So at least in the retina, it was definitely a great boon.
And just one simple question: is that did you use 2.5%? Or yeah, that's
yeah. So we used the 2.5%. That was volume to volume though, because of the way it was shipped to us. I believe it was at a gram per mil, or something like that. Yeah. So we had a 100% solution, so we did 2.5% volume to volume of the 2.3 to 1 SMA that we were given. We didn't start from powder or anything like that. We had the pre-activated SMA.
Excellent.